So we were discussing that amorphous form of drug is more soluble than crystalline form. So the polymorphic forms are those phenomena where the drug can exist in more than one form, either amorphous form and crystalline form or more than one crystalline form. Amorphous form is more soluble than the crystalline form. So consider this point for the competitive exams, that which form may be more soluble form. So it is the amorphous form which is more soluble than that of crystalline form. Example, in case of antibiotics, novobiazine, amorphous form of novobiazine is used in market. Novobiazine is available in crystalline form as well as amorphous form. Amorphous form is used because of its solubility. And then there is another phenomena which is known as pseudo-polymorphism. It is similar to polymorphism, but it is not exactly the polymorphism. That is the nature of the drug existing in different forms. That is, the drug can exist in anhydrous form as well as hydrated form. When the drug gets crystallined, in its crystalline lattice, it may associate some solvent molecules which has been involved in the synthetic process. That is known as solvate. So when it associates water as the solvate, then that is known as hydrate. So hydrate may be anhydrous form, monohydrate, dihydrate, decahydrate, octahydrate, like different hydrate forms are there. Now again the question arises, which form is more soluble form. So we might be imagining that hydrate form may be more soluble since it has water in it. No, it has water in its crystalline structure. So it has less solubility, whereas anhydrous form of drug is more soluble. Example, ampicillin available in anhydrous form as well as the trihydrate form. So generally anhydrous form of ampicillin is more soluble than that of this trihydrate form. So this is all about the polymorphism, pseudo-polymorphism, that is mainly in the crystalline nature of the drug. Depending upon its crystalline nature, and polymorphs which are soluble, stable, can be used for oral drug absorption. So another point in physicochemical properties of the drug in its solid form is drug stability in GIT. There are some drugs which have instability in GI region. Some drugs may be unstable in gastric environment, some has instability in intestinal region. Like example for those drugs is penicillin, digoxin. These all the drugs have instability in GI region. Penicillin is having gastric instability. Digoxin also has problem like acid hydrolytic and enzyme hydrolytic issue. So how we can administer these drugs orally? If you need to orally administer these drugs, either we can go for enteric-coated formulation or less soluble form of drug can be made, because highly soluble have more instability issue. So when you make less soluble form of drug, will have more stability, or otherwise we can go for prodrug. Prodrug may be stable in GI environment and it will release its active form of drug in site of action. So less soluble form can be used, or prodrug. Or another option is that you can choose the alternate route of drug administration. Instead of oral route, we can go for other routes. Like in penicillin, parenteral route is preferred over oral route since it has its GI degradation. So this is all about the factors that affect the drug's absorption and the drug is in its solid state. Thank you. We will discuss about the next, that is drug when it is in solution state, what all the factors that are going to affect in my next video. Thank you very much. So welcome to the next video, that is a series of factors affecting oral drug absorption. So we were discussing in our last video regarding the physicochemical properties of the drug, how those properties going to affect the oral drug absorption, especially in case of the drug when it is administered in solid dosage form. The solid dosage form must first of all undergo solution, dissolution, and then they have to get absorbed. So now the drug is in solution state. Now what all the factors which are going to affect the drug absorption? Already we discussed what all the factors influences the solid drug to convert into solution. Now what all the factors which will affect the permeation of this solution form of drug? So there are two factors which will affect this drugs PKA and GAPH, and the another is lipophilicity of the drug. So we know already the drug which are unionized form. So which form of drug is absorbed more? Unionized form of drug and lipophilic. Since unionized form is also lipophilic, these lipophilic molecules can easily pass through the lipophilic membrane. Since our membrane itself is lipophilic in nature, so easily drug molecules which are lipid soluble and which are unionized can easily permeate through this membrane, biological membrane. So this is explained by the pH partition theory. So the students have to note here for the exam point of view, this pH partition theory and its deviation may appear in short notes. Some 5 marks or sometimes even 10 marks also might be asked in case of pH partition theory. So the pH partition theory relates these two parameters, that is drugs PKA and GAPH and lipophilicity. Now, what is that pH partition theory? Depending upon the pH and the partition coefficient, the drug absorption occurs. So, the drug absorption is dependent on either GAPH or the partition coefficient of the drug. That is the meaning of the pH partition theory. So, we will move on to that pH partition theory later. Before that, we have to discuss about the first factor, drug PKA and GIPH. So, the property of the drug is PKA. Now, what is PKA? Like pH, PKA is the negative log of acidity constant. So, this is the value of drug which determines the drug's fraction of ionization and unionization. How much the drug have to be ionized and unionized, and where the drug have to get ionized, at with what pH the drug gets ionized, depend upon the pKa of the drug. Now the pKa scale also starts from 1 to 14. Unlike pH, the pKa of weak any acidic drugs will have the value from 1 to 14, and the pKa of basic drug also can go from 1 to 14, but it will be reversal side 14 to 1. So what does it mean? In pH we know that 7 is the neutral point. Below 7 the pH will be acidic and above 7 it will be basic. But not like that. In pKa 1 to 14 can carry the value for acids as well as for bases. What does it mean? So strong acid will have the value from 1 and it approaches to weak acid, whereas strong base will have value from 14 till weak base, that is the pKa may reduce up to 1. So the acidic and basic both pKa can be appear between 1 to 14. Now what is this pKa? It is the fraction of ionization and unionization of drug determining factor is known as pKa. So the weakly acidic or strongly acidic drug or weakly basic or the strongly basic drug where it gets ionized and where it gets unionized. It depends upon the GI pH. So we have difference in pH in GIT. That is, stomach has acidic pH and small intestine has basic pH and large intestine still basic pH. So the GI pH determines the drug's ionization. Acidic drug as well as basic drugs ionization will be determined by the GI pH. So we have three parameters: pH, PKA and drugs ionization and unionization. So these three parameters can be interrelated into a single equation which is known as Henderson-Hasselbalch equation. So what is this Henderson-Hasselbalch equation? The relationship which relates pH, pKa with that of unionized and ionized fraction of the drug. Now the Henderson-Hasselbalch equation for weakly acidic drug is pH is equal to pKa plus log of ionized fraction upon unionized fraction. Whereas for weak base, this equation will become pH is equal to pKa plus log of, this will be reversed, that is unionized upon ionized. So this is the Henderson-Hasselbalch equation for weak acid and weak base. Now what does it mean? pH is equal to pKa plus log of ionized upon unionized concentration of drug. That means for a weakly acidic drug, that means pKa of the drug is acidic in nature. Somewhere it exists in the weakly acidic region. So according to the pKa of the drug is weakly acidic. Now what happens when you place this weakly acidic drug in the pH of acidic pH, that is reduced pH? This is inversely proportional to unionized fraction and directly proportional to ionized fraction. That means when the acidic drug is placed in reduced pH, that is if the pH will be less, then the unionized fraction will be more. That is, acidic drug gets unionized in acidic pH, we know already, and acidic drug will be ionized in basic pH. That means if the pH is less where the weakly acidic drug is placed, that is when the weakly acidic drug is placed in stomach, that is in less pH, the ionized form of drug will be more, whereas unionized form of drug will be, that is when the pH is reduced, the ionized drug absorption will be increased, whereas when the pH is increase in pH will have increase in ionized fraction, whereas reduction in pH will have reduced ionized concentration. That means weakly acidic drug when it is placed in stomach will have unionized fraction more, whereas when the weakly acidic drug is placed in the intestinal pH, it will have ionized fraction more. See, when the weakly acidic drug placed in stomach, that is when there is reduction in pH, there will be reduction in the ionized fraction, unionized fraction. That is, pH is directly proportional to ionized form of drug and pH is inversely proportional to unionized fraction. So reduction in pH will reduce the unionized concentration, whereas increase in pH will increase the ionized concentration. That's it. Whereas in case of weakly basic drug, pH is directly proportional to unionized drug. That is, the pKa is weak base. When you increase the pH, that is when the weak basic drug is placed in intestinal medium, there will be increased in the unionized form of drug. We know already basic drug when placed in intestinal will have more unionized concentration, whereas basic drug when it is placed in acidic pH will increase the ionized concentration. This is the significance of Henderson-Hasselbalch equation, which in conclusion it explains that weakly acidic drug will have more ionization in stomach pH or acidic pH and will have more absorption. Weakly acidic drug absorbs more from stomach because these drugs are unionized in stomach pH. Whereas weakly basic drugs contrary. So this weakly basic drug will be absorbed more from intestinal pH because they will be unionized at that particular pH. That can be explained by this Henderson-Hasselbalch equation. So now, what is the significance of this PKA? So we discussed about how much the concentration will be ionized and unionized in case of GAPH and depending upon the PKA. Now what is the significance of this PKA with oral absorption? How the relationship between the oral absorption and PKA occurs? Now the drug, consider a weakly acidic drug whose PKA is greater than 8. If it is less than 8, it will be strong acid. So when the PKA of the weakly acidic drug is greater than 8. So what does it mean? PKA means it is the constant, or it is the index which explains the property of the ionization property of the drug. That is, when it is placed in different pH, the drug gets ionized at the pH 8. Before 8, the drug will be in unionized state in case of weak acid. See, in reversal case, that is, if the weak base will have the pKa around 8, this will have ionization after the pH 8. But after the pH, before the pH 8, they will be in ionized state. So acidic environment, they will be ionized state. So what does it mean? If the drug is having pKa greater than 8, that means the drug will start its ionization at pH 8. Before 8, that is from 1 to 7, the drug will be in its unionized state, and the ionization starts from the pH 8, 9, 10, 11, 12, 13, 14. Till 14, it will get ionized. It starts from 8. This is the meaning of PKA. So you consider the weak acid whose PKA is greater than 8, and our physiological pH, that is GI-pH, starts from 1 till 8. That is, stomach has pH around 1 to 3, and then 6 to 7, then 7 to 8. So GIT has pH of about 1 to 8. So when the weakly acidic drug whose pKa is greater than 8, when it is placed in such pH, that is in GI-pH, what happens? The drug gets absorbed, ionized after 8. But before 8, they will be in unionized state. Its ionization starts after 8. It is unionized between this pH 1 to 8. So throughout GIT, these drugs will be absorbed uniformly. These weakly acidic drugs like theophylline, example for the drug weakly acidic drugs whose pKa is between this, greater than 8, will have unionization throughout GIT, so gets absorbed throughout GIT. And the reversal case, in case of weak base, when first of all we consider the strong acid, then we discuss about the weak base. So the strong acid, example cromolyn, may have pKa around, like if you consider it as like 1, that means from 1 the ionization starts. pKa 1 means the ionization starts at 1. So between 1 to 8, the drug gets ionized. So throughout GIT, the drug will be in its ionized state. So absorption will be poor in case of strong acids, whereas in case of weak acids, the absorption will be uniform throughout GIT. Same in case of weak base. Consider weak base like diazepam, whose pKa is less than 5. So it is a weak base having pKa less than 5. That means less than 5 means towards 4, 3, 2, 1. So this weak base will be unionized between 1 and 8. That means throughout GIT they will be unionized. So they will be absorbed uniformly throughout the GIT. Whereas strong base, example guanethidine, they gets ionized throughout GIT and its absorption will be poor. So this is the significance of pKa and oral drug absorption. That is, when the drug is weakly acidic, this will be ionized throughout GIT and absorption will be uniform throughout GIT. Whereas in case of strong acid, they will be ionized throughout GIT, so the absorption will be poor throughout GIT. Whereas in case of weak base, they will be again unionized throughout GIT and absorption will be greater. Whereas the strong base have ionization throughout and the poor absorption occurs in case of strong base. That is why weak acids and weak bases are preferred for drugs instead of strong acid and strong base. Now we will discuss about the next factor, that is the property of the drug, lipophilicity. Lipophilicity. So partition coefficient explains the lipophilicity of the drug. So the index used to determine the lipophilicity of the drug is its partition coefficient. So greater the partition coefficient, more the lipophilic the drug is. Lesser the partition coefficient, hydrophilic the drug is. So the drug must be lipophilic in nature. If the drug is polar in nature, in case of drugs like insulin, heparin, gentamicin, they have poor absorption since they are hydrophilic in nature, which have less lipophilicity. That is, they are more hydrophilic since they are polar in nature. So they cannot be administered through oral route. They are preferred through parenteral route because their oral absorption is very less. So what we can do to improve the lipid solubility if the drug is not lipid soluble in nature, when the drug is polar in nature? Either you select the other route of drug administration or you can go for structural modification of the drug. That is, the structure which is having the less lipophilic nature can be converted into a high lipophilic structure, or otherwise we can go for a prodrug formulation with high lipophilicity. So structural modification, for this example, we have the antibiotic lincomycin that can be converted into clindamycin. This lincomycin is polar in nature since they have hydroxyl group in their structure. This clindamycin can be prepared by replacing this polar group by lipophilic group chlorine. So substitution of chlorine instead of OH group will convert the hydrophilic moiety into lipophilic. Thus we can enhance the lipophilicity of the substance, or otherwise we can go for a prodrug formulation. Example, chlorazepate is the prodrug of nordiazepam. So you can make the prodrug form which is highly lipophilic, can enhance the permeation of the drug through membrane, that can permeate through the membrane, and once it reaches the site of action, they may convert into its original active form of drug. This can be done for this lipid solubility. So this is all about all physicochemical properties of the drug that are going to affect the oral drug absorption. So we discussed when the drug is in solid form what all the factors which are going to affect, and we discussed today about the factors when the drug is in solution form. Just two factors, drugs PKA and GAPH, and second factor was lipophilicity of the drug. These two we discussed today, and in my next video we are planning to discuss about the pH partition theory and its deviation. It again comes under this factor, that is drug when it is in solution form, what are the factors. So combining these three parameters, drugs PKA, pH and the lipophilicity, we can explain by pH partition theory. So again this will be taking another 15 to 20 minutes. So I'll just make a separate video for that pH partition theory. Thank you.